So tissue is one of the critical issues in any rare disease, especially rare types of cancer. Because cancers are, because the way that cancers begin is as a unique event in certain cells in a person, that person does not carry any of the molecular important findings of the disease once the disease has been removed. So I know that sounds a little circuitous, but cancer is not essentially a genetic disease where we carry the genes that cause it through our whole bodies most of the time. There are exceptions to that in a few familial types of cancer. But clear cell sarcoma is virtually never associated with a familial cancer type. And so that means that the only place where the specific molecular drivers of that cancer are present is in the cancer cells themselves. Those cancer cells, once they've been radiated, once they've been treated with aggressive chemotherapy, once they've been removed and put into pathology fixation solutions, they are in many ways no longer useful for the types of research that we can do on fresh tissue. They are still important, and they still can be archived in some of those other states if they've already been through the pathology solutions. That's okay. We still would like to have those kinds of tissues collected and assembled into a set, basically, so that we can not only have clinical data from patients, but we can have actual tissue data and all the data we can derive from that tissue in terms of stains that we can use on it by immunohistochemistry, which is a way of testing for a specific protein in a tissue, or some of the molecular tests that we can do on even tissue that has been processed through formalin and embedded in paraffin, which is what happens to tissues when they're treated by pathologists most of the time. Even these tissues can be used to study the molecular drivers of clear cell sarcoma. And so even a registry or a bank of those kinds of tissues can be very, very important. But what would be even better is anyone who can connect with Sarah's Cure and be able to make sure that their tissue is processed appropriately from the beginning. That's even better. I mean, any time when we can get fresh frozen tissue samples, which many hospitals are able to do if they know to do it before the surgery, that can be very critical for any type of research can use that kind of tissue. It's invaluable. I would have to say it is one of the biggest limitations in something like clear cell sarcoma. So I recently completed a study with trying to understand the molecular drivers of clear cell sarcoma other than the main driver, which we think of as this EWSR1 ATF1 fusion gene. But there are other drivers that we can detect by looking at samples. And we were only able to put together a series of patients from samples that were basically dating back 20 years and have been embedded in paraffin by a very wise pathologist who was basically collecting these cases over many years as they were sent to him in consultation. Every case he would save a few slides that were unstained. And we were able to use these tissues to try to understand some of the very basic drivers of clear cell sarcoma. However, we would have much preferred to have had frozen tissues if we could have had even more than 2 or 3, which is about all that we could find at the time. So I think a tissue banking effort or an effort to register tissues up front for patients, any patients who actually connect with Sarah's Cure before they have their surgery, that would be an invaluable resource.